Very good morning, students. Welcome to biology classes. Today we are going to see 11th science biology and we are going to see the next topics of chapter number 14, that is respiration in plants, right? So already we have covered the introductory part of this chapter. So today we are going to resume this chapter with the next topics, upcoming topics, which are one of the most important topics of this chapter. So in the previous class, we have seen about the introductory part, and today we are going to see about the aerobic respiration, right? So let's just recall it that respiration has been divided into two parts, that is they are of two types. First is aerobic and second is anaerobic. Aerobic respiration is the respiration which takes place in the presence of the oxygen, right? And anaerobic respiration is the respiration in which the oxygen is not involved, that is in the absence of the oxygen. In the aerobic respiration, we are going to see three types of pathways. The first is glycolysis, second is Krebs cycle, and third is the terminal oxidation, right? So let's start with the aerobic respiration. So aerobic respiration is an enzymatically controlled release of energy in a stepwise catabolic process of complete oxidation of organic food into carbon dioxide and water with oxygen acting as terminal oxidant. So let's go and understand this definition words wise, right? Term wise. So aerobic respiration is enzymatically controlled. That means this reaction or this respiration has been controlled by the enzymes, right? Where the release of energy takes place in a stepwise manner, right? So when the release of energy takes place with the help of the enzymes in a stepwise manner, and where the catabolic process of complete oxidation, that catabolic means the breakdown, right? So the catabolic means the breakdown of the complex molecules takes place, that is the complete oxidation takes place of the organic food, and it converts those complex uh, complex molecules into simple forms, that is carbon dioxide and water. And this process, that is the oxidation of the organic food, the conversion which is taking place, or the breakdown which is taking place, this is occurring in the presence of the oxygen, right? And for this reason, this type of respiration is known as aerobic respiration. Now in the aerobic respiration, the first pathway which we are going to see is the glycolysis, right? The scheme of glycolysis is given by Gustav Embden, Otto Meyerhoff, and J. Parnas. It is also called as EMP pathway. These are the scientists who proposed this model, or who proposed or established this glycolysis, and it is also known as EMP pathway. This question can be asked as that simply as glycolysis, or explain the EMP pathway, right? So this way the glycolysis has been given. Now glycolysis is the partial oxidation of glucose, right? So here what happens, the glycolysis is what the partial oxidation of glucose takes place, or similar hexose sugar, into two molecules of pyruvic acid. And this occurs through a series of enzyme mediated reaction releasing some ATP and NADH2, and this occurs inside the cytoplasm, right? So first thing is the glycolysis is the conversion of the glucose, right, to the pyruvic acid. So the molecules of glucose are converted into two molecules. So one molecule of glucose is converted into two molecules of pyruvic acid, and this conversion takes place, or it occurs in a series of enzymes mediated reactions. That means there are at each and every step the enzymes are involved, and with the help of the enzymes, right, they are releasing some ATP as well as the NADH2, right? And this process is taking place inside the cytoplasm. So let's see about the glycolysis. So here you can see this is the whole flow chart of the glycolysis which you need to learn by heart. Now, the first thing that we are going to see here is the conversion of the glucose molecule into two molecules of pyruvic acid. So here at the top, you can see over here the glucose, which is a 6-carbon compound, right? This 6-carbon compound is converted into two carbons of pyruvic acid, which are 3-carbon compound, right? So this 6-carbon is split into two molecules of pyruvic acid, which are 3-carbon compound. So let's see here. There are so many different steps, and these steps are needed to be followed in order to reach this step, right? Or we can say the last step. The first step is the conversion of the glucose, which is a 6-carbon compound, to glucose 6-phosphate. So here what happens, the glucose, right, this glucose is being converted to glucose 6-phosphate, and here ATP energy is used. So one phosphate has been donated, and for this it is been used, right? For this reason, as this phosphate is entering into the glucose molecule, for this reason on the 6th position, the phosphate molecule is added. For this reason, this glucose gets converted to glucose 6-phosphate. But again, just as the glucose, which was a 6-carbon compound, this glucose 6-phosphate is also a 6-carbon compound. So here is the first conversion where the ATP energy is being utilized, and this ATP, upon uptaking the phosphate molecule, it gets converted to ADP. The full form of ATP is adenosine triphosphate, while ADP is adenosine diphosphate. So as here one phosphate is being used, for this reason from tri it is converted to di, right? So this is the first conversion, that is glucose gets converted to glucose 6-phosphate. Both of them are here in a 6-carbon compound. Now this glucose 6-phosphate gets converted into fructose 6-phosphate, right? See here the enzyme which is involved in the first step, right? That is hexokinase, right? So kinase is the enzyme which is responsible for utilizing the energy, and it is providing the phosphate molecule, right? So here the conversion of the glucose to glucose 6-phosphate takes place in the presence of the hexokinase. So this is the first enzyme which is responsible for the first step, that is the glucose to glucose 6-phosphate. Now, the second step is glucose 6-phosphate, which is a 6-carbon compound. It gets converted to fructose 6-phosphate, which is again 6-carbon compound. Just here, see glucose as well as fructose. Glucose and fructose, they both are isomers of each other, right? As isomers are there, for this reason here in the second step, the enzyme which is known as isomerases is being involved. So here for the conversion of glucose 6-phosphate to fructose 6-phosphate, enzyme is involved which is known as isomerase, right? So this isomerases is responsible for producing the isomer. So from the glucose, it forms its isomer, and that is fructose 6-phosphate, right? So none another, they both of them are same, but just the structure of it is different. Now again, in the third step, when the fructose 6-phosphate, that is a 6-carbon compound, it gets converted to fructose 1,6-bisphosphate, and this is also going to be the 6-carbon compound. Now I would like to uh, draw your attention over in this step where this fructose 6-phosphate, that is the phosphate is right now present on the 6th position, right? But here again one phosphate is being utilized, or it is being used, and for this reason that again another phosphate is being added on the first position. So in the fructose structure, on the first position again one phosphate is being added. So here now on the first position as well as on the 6th position, two phosphates are there, and for this reason two phosphates are there. For that, here it is written as bisphosphate, right? So here the conversion of this says that initially the fructose 6-phosphate, right, here only one phosphate was there, and this phosphate was on the 6th position, right? But here after adding of the another phosphate or utilizing for ATP energy, again one phosphate is being added into the fructose 6 phosphate, and for this reason, as another phosphate is added on the first position, right? For this reason, two phosphates are there and it is said to be bis phosphate. But here again, this fructose 6 phosphate was also 6 carbon compound, and this fructose 1,6 bis phosphate, this is also a 6 carbon compound. Now here the main tragedy takes place, right? And here what happens is this 6 carbon compound, it gets splitted into two molecules, and that will be 3 carbon and 3 carbon compound. But these two of them are different with from each other, right? So the first is this fructose 1,6 bisphosphate, that is the carbon that 6 carbon compound, right? This gets converted to the first molecule, that is the triose phosphate, right? So here what happens, this fructose 1,6 bisphosphate gets converted to triose phosphate, which is simply known as glyceraldehyde 3 phosphate, right? So here again there will be our 6, here the division is taking place at 6 from 6 carbon, 3 carbon is separating and 3 carbon is separating, right? So the first is which is formed, that is glyceraldehyde 3 phosphate, right? So this is the first thing, and the second which is forming, that is where I'm showing the cursor. Again, that is a triose phosphate, but that will be DHAP, that is dihydroxyacetone phosphate, right? This is also a 3 carbon compound. So the fructose 1,6 bisphosphate, which was a 6 carbon compound, it gets split into two molecules of 3 carbon compounds. The first is the glyceraldehyde 3 phosphate, and second is DHAP, that is dihydroxyacetone phosphate, right? Now this triose phosphate, that is the dihydroxyacetone phosphate, here you can, where I'm showing the cursor, this orange colored reaction, it shows the reversible reaction. That means this that DHAP is or it has the ability to form the glyceraldehyde 3 phosphate. So none another, as this whole sequence is running, this DHAP is getting converted into glyceraldehyde 3 phosphate, right? For this reason, already we have one glyceraldehyde 3 phosphate, and another glyceraldehyde 3 phosphate is being formed upon the conversion of DHAP into the glyceraldehyde 3 phosphate. So now we will be having two molecules of glyceraldehyde 3 phosphate, right? So I hope you are very clear up till this. Now what happens over here? Now here, from here you can see over here 2 into is written, right? That is 2 multiplied by, as one is going to as one product is going to be formed from the glyceraldehyde 3 phosphate, and second product is going to be formed from the second molecule, that is DHAP, right? But this DHAP is already going to be converted into glyceraldehyde 3 phosphate. So two molecules are there. For this reason, after this step, two cycles are running, right? So here now what happens is here this glyceraldehyde 3 phosphate, it here the NAD plus, this gets converted to NADH plus H plus. That is, here the reduction takes place, and for this reason here the H plus is being formed, right? So the H is being utilized, and for this reason this NAD gets converted to NADH plus H plus. So with this glyceraldehyde 3 phosphate gets converted to 2 into triose bisphosphate, right? This triose phosphate which was there, this triose phosphate gets converted into triose bisphosphate. Why? Because here two phosphates will be present, right? So already there was presence of 3 phosphates, and along with that 3 phosphates, another 2 phosphates are being added. So the protons are being added, and for this reason, this triose phosphate gets converted to triose bisphosphate, right? This glyceraldehyde 3 phosphate gets converted to 1,3-bisphosphoglyceric acid. So here on the first and on the third positions, right? So on the in this triose bisphosphate structure, right? What happens? on the first position and on the third position, two phosphates are being added, and for this reason, this compound, which is a 3-carbon compound, is known as 1,3-bisphosphoglyceric acid, right? Now, so here the reduction took place and the protons were removed, right? Now again over here, see, after the conversion of this particular triose phosphate to triose bisphosphate takes place, after that, here the energy is not utilized, but the energy is formed. So here the adenosine diphosphate, so here what happens? Now already we have the phosphates, right? So here what happens? The bisphosphate, so from bisphosphate, that means two phosphates are there. So from this two phosphate, one phosphate is being one phosphate is released out, and this phosphate is being used in the formation of the ATP energy, right? So this ADP gets converted to ATP, that is di gets converted to tri upon releasing of one phosphate from this particular step. So the triose bisphosphate gets converted to triose phosphate, right? So this triose phosphate, that is from two phosphate, only one phosphate is left because one phosphate is being removed out, right? So this 2 into triose bisphosphate gets converted to triose phosphate. Again, it will be 2 in number, right? So this 1,3-bisphosphoglyceric acid gets converted to 3-phosphoglyceric acid, right? So from the first position, the one phosphate is been removed, and that is utilized in the formation of ATP energy. And hence this 1,3-bisphosphoglyceric acid gets converted to 3-phosphoglyceric acid. So only it is left on the third position. Now this triose phosphate gets, or we can say the 3-phosphoglyceric acid, which is a 3-carbon compound. This 3-phosphoglyceric acid gets converted to 2-phosphoglycerate, right? So 2-phosphoglycerate, as again over here, one phosphate has been released out, right? And here the changes which are taking place is what? From the third position, the phosphate is being the phosphate which was present on the third position, it is changing its position and it is going on the second position. So from third position where the phosphate was present, right? Now in the 2-phosphoglycerate, on the second position, the phosphate is present. At this step here, there will be a removal of the H2O, that is the water molecule. So upon the release out of the water molecule, this 2-phosphoglycerate gets converted to 2-phosphoenol pyruvate. So upon the release out of the water, this phosphoglycerate gets converted into 2 into phosphoenol pyruvate. And here in the last step also, what happens? The phosphate is being removed out, and for this reason, that removed phosphate is useful in the production of the ATP energy. So from this phosphoenol pyruvate, the phosphate is being removed. So already only one phosphate was left. So that one phosphate is also now removed out, and for this reason, this phosphoenol pyruvate gets converted into pyruvate acid. And here two, that means as two chains are running, or two flowcharts or two molecules are running side by side, for this reason we are getting two molecules of pyruvic acid at the end of the glycolysis, and this is a 3-carbon compound, right? So this is the whole glycolysis where the conversion of the glucose, that is a 6-carbon compound, takes place into the two molecules of pyruvic acid, which is a 3-carbon compound, right? So in plants, the glucose is derived from sucrose or from storage carbohydrates. So here what happens? In plants, what happens? The glucose is being derived, that is, it is being formed from the sucrose, or it is we are going to get, or the plants are going to get from the carbohydrates which are stored, right? This sucrose is converted into the glucose and fructose by the enzyme invertase. So as I said over here, in the second step, the glucose 6-phosphate was converted into fructose 6-phosphate, as these two of them are the isomers of each other, right? Here same way the sucrose is converted into glucose, and the fructose. So this sucrose is converted into two types of sugar. The first is glucose and the second is fructose. And this type of conversion is taking place by a specialized enzyme which is known as invertase, right? So this is all about the steps which we have already seen in the glycolysis, right? So I hope you are very clear with the first type of the aerobic respiration pathway, and that is glycolysis. I hope you are very clear, and I hope to see you again. Thank you.